Welcome everyone to Atara Biotherapeutics company presentation at the ARM meeting on the MESA 2021. After the usual forward-looking statement, I would like to highlight the important mission that Atara Biotherapeutics is focused on, which is to transform the lives of patients with serious disease through pioneering science, teamwork, and a commitment to excellence in leveraging our allogeneic T-cell immunotherapy platform there. We are a leading company in that space in a sense that we have a very differentiated allogeneic T-cell therapy platform that relies upon EBV for Epstein-Barr virus T-cells and technologies, especially next-gen CAR-T technologies, to develop multiple allogeneic cell therapy. We have with Tab-cel a true first-in-kind, the most advanced allogeneic T-cell immunotherapy today, which is in phase 3 and for which we expect to complete a European submission in November 21 and have a BLA submission completed in Q1 of 2022. This is again a late-stage oncology program with breakthrough designation in the US and prime designation in Europe that is really trying to address a very significant unmet medical need. Then in the clinic, we have also ATA188. This is potentially transformative treatment of multiple sclerosis, which is right now in randomized controlled trial with placebo-controlled data expected over the next few months, which could really address an important need of patients, but also lead to partnering opportunities and pivotal studies. The third pillar of our activity is around next-gen allogeneic CAR-Ts with a significant competitive portfolio of assets there, and one in particular related to mesothelin, an important target for solid tumors that has been validated by a collaboration with Bayer, and for which the first program is already in a clinic. All of these assets are really supported by proven technical capabilities around process science that allows us to develop a true platform that allows to treat a wide range of either EBV-associated disease or other hematological and solid tumors through engineered CAR TCR. We take cells from healthy donors, we're able to select and manufacture these as EBV T cells, allogeneic EBV T cells that can address EBV-associated disease. And we have two products there, Tab-cel and ATA188, that are different products depending on the antigens that they are targeting. But we can then also develop for next-gen CAR specific allogeneic CAR-Ts that could have not only the ability to target specific liquid and solid tumors, but also to retain the benefit of EBV T cells. And when we're talking about the benefit of EBV T cells, we really have in mind our vision, which is to transcend the limitation of current cell therapy by harnessing the power of this EBV T cell from healthy donors. We've already been proving through clinical studies as well as preclinical studies that we can address with this platform based on EBV T cells aspects related to the need for durable efficacy, for persistence of the cells, for tolerability. And this is again proven clinically. We also have shown that we can scale up our manufacturing to a very significant level, and we have a truly flexible off-the-shelf therapy. We've experience now in supplying our allogeneic T cells to hospitals around the world within just a few days from our inventory, and we have excellent patient access and experience through this clinical program. So main differentiation here is that we leverage this benefit of EBV T cells, but it's already proven and optimized and validated at the clinical stage. Now, this is applied to our portfolio of assets. And here we have on this slide, the different key assets with Tab-cel that I already mentioned, ATA188 in randomized controlled trial in phase 2 in progressive MS, and then a number of programs of CAR-T targeting mesothelin, CD19, and also some various B-cell malignancies and solid tumors at the earlier development stage. The first program, of course, Tab-cel, is addressing the real issue of EBV-driven cancer, because EBV, Epstein-Barr virus, cause rare and serious cancer in patients with impaired immune function. And here with this product Tab-cel, we're able to boost the immune system of the patient in bringing them this additional immunity so they can fight against this EBV-driven cancer. We are now in phase 3 in previously treated EBV-positive PTLD for post-transplant lymphoproliferative disorder. And we're also in a phase 2 multi-cohort study underway, covering 6 additional patient populations with the aim to expand the label of Tab-cel there. Tab-cel is truly specific. It specifically targets and kills EBV-infected cells, addressing really this disease at the source. Now, we have already data coming from our phase 2 program, EAP, as well as an interim analysis of our pivotal study that clearly show and demonstrate high and durable treatment effect. We have objective response rate of 50% and over. We have over 80% survival at 2 years in responders in our phase 2 and EAP program, whereas this patient without treatment will have an overall survival, median overall survival of just 2 to 3 months. And this is being achieved with an excellent safety profile, with no CRS or neurotoxicity observed, no treatment-related mortality, and a low burden of administration for the healthcare system. Is just a 5 to 10 minutes IV, no pretreatment is required, and just a 2 hours monitoring, allowing to treat patients even when they are outpatient. And this could be delivered within 3 days to any hospital in need, and that's a truly off-the-shelf allogeneic T-cell therapy advantage there. So as you can see, there is a compelling value proposition for EBV-positive PTLD patient and healthcare system with Tab-cel. And we're looking forward for filing and launching this product in 2022. Now, our second product is addressing also a very important unmet medical need in multiple sclerosis, in particular in progressive MS, where the prognosis of the patient today is very poor. The current therapies are just modestly delaying progression, but none of them is fundamentally altering the progression of the disease. And we are developing our product AT188 in that space because there have been growing evidence that Epstein-Barr virus latent infection has a major role in the pathogenesis of MS. Not only that prior EBV infection is necessary for a patient to develop MS, but MS may be mediated by B cells that are infected with EBV in a CNS. Now, our aim here is really to develop a transformative therapy in progressive MS and other type of MS. And why do we call that a potential transformative therapy? Because current treatment is just delaying the progression by a few months, where here our aim is either to halt progression or even more so to lead to a sustained reversal of disability. We believe that AT188 has the potential to be this transformative treatment of MS because we believe it's addressing the underlying biology of the disease in the sense that MS pathogenesis is increasingly linked to EBV latent infection. And we have really a precision targeting that limits off-target activity to really kill and hunt and kill only the B cell and plasma cell that are infected by EBV and that we believe are the trigger of the progression of symptoms in MS. We know our T cells are penetrating the CNS, they traffic and cross the blood-brain barrier, and we've already observed sustained disability improvement and acceptable safety profile in our phase 1A. Here on this slide, you have some of the results we presented last year that clearly show that, especially at high dose, we have a significant number of patients with sustained disability improvement, with up to 50% sustained disability improvement in the high dose cohort at 15 months. And this is also compounded by the fact that patients achieving SDI have more improvement in different patient-reported outcomes of importance for MS patients there. So very encouraging, very exciting data on MS. Now, the third pillar of our activity is around leveraging our EBV platform to optimize CAR and TCR therapies. And here we're doing so for various next-gen technologies. We're working on specific targets and also multi-targeted CAR with dual targeting and gating. We're also working on inserting a next-gen co-stimulatory domain, particularly 1XX developed by Michel Sadler at Memorial. And these novel co-stimulatory domains are here to offer less T-cell exhaustion, leading to longer functional persistence, which is so important in CAR-T field. We also are able to armor these CAR-Ts in adding things like features like PD-1 dominant negative receptors that can provide intrinsic checkpoint inhibition to unlock the solid tumor microenvironment. So here we have two advanced programs. One is targeting mesothelin, and that's being developed in collaboration with Bayer and, of course, Memorial Sloan Kettering. And here we address mesothelin as a very well-established target associated with aggressive solid tumors, mesothelioma, lung cancer, ovarian cancer, and pancreatic cancer. And here we have a very unique design of the CAR-T with this new costimulatory domain as well as a PD-1 DNR. And we've proven in a number of preclinical studies that were already presented that this is associated with less cell exhaustions, improvement in functional persistence, serial cell killing, and enhanced in vivo efficacy when compared with first-generation CAR-Ts there. So we have a program called ATA2271 that is already in the clinic, which is an autologous version of the product, and we are accelerating the development of the allogenic version called ATA3271, which is this off-the-shelf allogenic EBV mesothelin CAR-T, for which we plan to file an IND in second half of 2022. The last program in advanced stage is ATA3219, where here we're developing the potential best-in-class off-the-shelf allogenic CD19 program with an IND expected in Q1 22. And this program is benefiting from this new costimulatory domain, from the EBV T cell, from healthy donors, but also supported by a very exciting clinical proof of principle that was presented last year by a team at Memorial, showing that having allogenic EBV CD19 CAR-T could allow to have a very high response rate, up to 83% CR in patients with advanced B-cell malignancies, together with long durability of response. These patients were followed for more than 2 years, 26.9 months exactly, and they were maintaining their CR at that stage of median follow-up there. So very exciting type of high efficacy, long durability of response, and at the same time, acceptable safety with no dose-limiting toxicity, no CRS or toxicity above grade 2. Very exciting clinical proof of principle. Now we are accelerating the development of our own Atara product, ATA3219, in that space. As you can see, over the next 15 months, there is a lot of progress across multiple fronts. We expect to be really into a situation having the first allogenic T-cell immunotherapy on the market in 2022. We also expect to have disability improvement data from the randomized control trial with ATA188. That has the potential to unlock a multi-billion-dollar opportunity. We have multiple programs on the CAR-T front with clinical data coming progressively in solid and liquid tumors. And of course, we are continuously scaling up and improving our platform, which is really the most advanced allogenic T-cell platform today. On this note, I would like to conclude in thanking you very much for your attention.